Hi, my name is Hannah Rogers. I'm from Milligan College, which is in East Tennessee. And for my project, I did the synthesis of unnatural dipeptides against Pseudomonas aeruginosa. This isn't a new project. Previous students from last semester, last year, started this project with our mentor professor, Dr. Kristen Mudrack, and my lab partner and I continued this project this semester. So why is our project important? First off, Pseudomonas aeruginosa is a very prominent bacteria in patients with what's called cystic fibrosis. CF is a genetic disorder where there's a mutation on what's called the CFTR gene, which stands for cystic fibrosis transmembrane receptor, which is on chromosome 7. What this gene codes for is a protein channel that allows chloride ions to be transported in and out of the cell. When this is messed up, that causes a lot of problems, and that's where CF comes in. There is a common misconception that CF is only a lung disorder, when actually it's a whole body disorder. It affects, as you can see in this figure, your sinuses, your skin, your liver, your pancreas, your intestines, and your reproductive organs. We just tend to focus on the lungs. Also, as you can see in this figure, a normal airway like mine has this thin liquidy layer of mucus, which is normal. But this is not the case in an airway with cystic fibrosis. As I said earlier, the membrane channels that are affected don't allow chloride ions to go where they're supposed to go, and other ions compensate, and this causes water to leave the airway, and it leaves us with this dehydrated, sticky, thick mucus, which is a breeding ground for bacterial infections. And that's where Pseudomonas aeruginosa comes in. As you can see in this graph, Pseudomonas aeruginosa is one of the top bacterial infections in patients with CFs, especially in preteens or older. And according to the Johns Hopkins Cystic Fibrosis Center webpage, 80% of CF patients have a chronic infection of Pseudomonas aeruginosa. So this project is really important. So another reason why this project is really important is due to antibiotic resistance. So as you can see in these figures, tobramycin and aztreonam, and one that's not here is casein, are common antibiotics used to treat Pseudomonas aeruginosa. But the problem is that this bacteria has adapted and mutated to where it isn't killed anymore by these antibiotics. So there's this urgency that we need new antibiotics, and that's why we chose to do this project. And we chose dipeptides. So what is a dipeptide? The dipeptide is when there's two amino acids put together with a bond, specifically a peptide bond. And so why dipeptides? Like, why should we choose that? There's two main reasons. One is because Pseudomonas aeruginosa is a gram-negative bacteria. As you can see in this figure on the left, it has a negative charge around it. For the dipeptides, in certain contexts, the amino terminals and the amino ends have a positive charge, as you can see in that figure. And as most of you know, negative and positive are attracted to one another. Another reason why dipeptides are a good choice is because in a bacteria, there's a cell membrane, and it consists of what's called a lipid bilayer. In our dipeptide, they have certain components that like lipids, and these are the ones that are pointed to by the blue arrow. They like lipids, so they can penetrate the bacteria very easily and cause the bacteria to lyse. So how do we make these dipeptides? First, we use what's called solid phase peptide synthesis, and we use what's called a resin, or in other words, a bead. And the reason why a resin is important, because it's easier to build up our products, to make our product with the bead present. And that's all this first step is doing. It's just preparing that resin. For the left figure and the middle figure, we use these vials to build up our product. And the right figure is these little containers that we put our final product in. So the second step After we prepare our resin and put them in the vials, we use what's called alkylation. So what this is basically doing, like I said earlier, dipeptides have 2 amino acids. We're making our first amino acid. And on the right side of the arrow, you can see what's called R1, and that's the group that we're adding. And the group that we're adding is fluorophenylalanine. And with the arrows, you can see that we changed up the position of the F or the fluoro to see if that made a difference in our results. So I did 2 fluoro and 3 fluoro here. Our third step is hydrolysis. So what that does is basically you can see on the left side of the arrow, there's groups that have pHs. We're just removing those and creating an amino group just so we can add our second amino acid. That's all that's doing. Our fourth step is acylation. And this is adding our second amino acid for our dipeptide. And as you can see on the right side of the arrow, it has R2, and that whole group that it's attached to, that's what we're adding. And I chose 3 amino acids, different amino acids. And the first one is alanine, then valine, and then D-valine. And what this is basically testing is there going to be a difference in how the bacteria is killed with 2-fluoro versus 3-fluoro? Is there going to be a difference between alanine, valine, and D-valine? For valine and D-valine, if you're not familiar with organic chemistry, it has to do with what's called stereochemistry. And what that basically is the spatial arrangement of a compound. So with valine, it has a wedge, which means it's facing towards you. D-valine has a dash, which means it's facing away from you. Is there going to be a difference with the stereochemistry on how it kills Pseudomonas aeruginosa? And our last step is removing the resin so we can continue with our crude product. So the next step is purification. So we take our crude product and we put it in a column, which is on the left side of the PowerPoint slide. And we would run our crude product through this column and get fractions of our pure product. And how we figured out where our pure product was is we used what's called a TLC plate. On the right, you can see a TLC plate of my crude products. And you see the little dots? That's just showing you where our crude products are on the TLC plate. So if these dots match up with the pure product TLC plate, then we know, hey, this fraction has the product in it, and we pull those together in one container to use in our testing. So our final step is a biofilm, and what this basically does is we're testing our products with the bacteria. So the D and E rows have the bacteria and my crude versus pure products, and you can see those listed on the right in purple and blue. And the F row here are my controls, and the only one that doesn't have Pseudomonas aeruginosa is F1, and that's just the media to see if it grows there. So why is it purple? Why are these wells purple? Well, it contains what's called crystal violet, and crystal violet is important because it sticks to bacteria very easily. So the higher amount of concentration of crystal violet in each well means that there's more bacteria present. And I measured this using what's called a UV vis. And in this chart, it kind of just shows my results visually here. In the x-axis, there's the concentration of crystal violet. And on the y-axis, it's the absorbance, which is measured by the UV vis machine. In blue, you can see A1 and B1, which is alanine. So it had the most concentration of crystal violet, which means it did not kill the bacteria that very well. In yellow, you can see A2 and B2, which is valine, which it also didn't kill it that very well. And then you can see with A3 and B3 in the pink, that is D-valine, and it killed Pseudomonas aeruginosa relatively well. And in orange, you can see my controls. And the Aeruginosa has a higher concentration of crystal violet, and I think that's because it wasn't concentrated enough to kill the bacteria, so it wasn't that great of a control. And also, as you can see, some of the controls have a little bit of concentration of crystal violet, and I think that's just because it's hard to remove all the crystal violet from the cuvettes, which we measure the absorbance in. So I made a chart of my significant results from my biofilm assay, and I use a one-tailed t-test for 2 independent means. So basically what all these numbers mean is that it shows the significant results using stats that D-valine was significantly better than alanine. So D-valine was the best amino acid chosen for killing the bacteria. And then what also this chart shows is that when it came to D-valine, 3-fluorophenylalanine was better than 2-fluorophenylalanine. So, and we can see this with the low p-values and higher t-values. So what does this mean? What does this mean for next steps? Well, it adds to previous results from Kelly Guerin and Andy Lamb. So in their paper, they talked about how the D stereoisomers killed Pseudomonas aeruginosa better than the L stereoisomers. And see, they didn't use the UV vis, so maybe we could redo theirs to see the actual concentration of the crystal violet. And then also in their paper, they talked about how 2-fluoro was better than 4-fluoro when it came to killing the bacteria. And in my results, 3-fluoro was better than 2-fluoro. So we need to do more trials to ensure significance. Is 3-fluoro the best position? Also, we need a better purification process because there wasn't a significant difference between the crude and pure products. And they were close in concentration, which is great. That means they are doing similar things, but maybe there's a way we can make our pure product even better. Also, another student who has started to be a part of this project is going to add quorum sensing. And quorum sensing is the way bacteria communicate with each other. So maybe they can include that in how they make their antibiotic. And that's the end of my presentation. If you have any questions, feel free to email me or message me. Thank you.